Hey guys, welcome to Pharma Catalyst. Today we'll discuss the interview questions related to pre-formulation studies. So let's start. Question 1. What is pre-formulation? Pre-formulation is the first step in the drug development where we assess the physicochemical properties of the active pharmaceutical ingredient. It helps us understand how the drug behaves alone and with excipients, ensuring that we select the right combination to create an effective and stable dosage form. This phase also helps in minimizing risks in the later stages of development. By gathering the important data, we can reduce both the time and the cost involved in the formulation processes. Next, what are the objectives of pre-formulation studies? The primary objective of pre-formulation studies is to collect detailed data on the drug's physical, chemical, and mechanical properties to facilitate the successful design of a dosage form. This includes selecting the right excipients and determining the optimal conditions for drug stability. Another goal is to ensure the drug's bioavailability by studying its solubility, permeability, and dissolution characteristics. These studies also help us understand the potential challenges in the drug formulation and addresses them early in the process. So the next question is, what are the types of pre-formulation studies? Pre-formulation studies can be categorized into physical, chemical, and biopharmaceutical studies. Physical studies assesses characteristics like particle size, solubility, and polymorphism. Chemical studies focuses on determining the drug stability, pKa, and degradation patterns. Biopharmaceutical studies look at the drug's permeability and dissolution rate, which are essential for understanding how the drug will perform in the body. Next question is, why is solubility important in pre-formulation? Solubility plays a critical role in drug absorption because a drug needs to dissolve before it can be absorbed into the bloodstream. Poor solubility can lead to low bioavailability, reducing the drug's effectiveness. Pre-formulation studies help us determine whether we need special techniques such as solid dispersion or micronization to improve solubility. This data also helps us decide the most appropriate dosage form for the drug. Next question is, what is polymorphism and why it is important? Polymorphism refers to ability of drug to exist in multiple crystalline forms, each with different physical properties. Different polymorphs can have varying solubility, stability, and bioavailability. Identifying and controlling the polymorphic form is important to ensure consistent drug performance. Choosing the right polymorph can significantly impact the drug's shelf life and therapeutic efficacy. Question 6. What is partition coefficient and its role? The partition coefficient or log P measures the lipophilicity of a drug by comparing its concentration in oil and water phases. This value helps predict the drug's absorption across biological membranes. Drugs with a log P value between 1 and 3 are generally ideal for oral formulations as they strike a balance between solubility and permeability. It is a critical parameter for assessing the drug's pharmacokinetic properties. Next question is, how is pKa relevant in formulation? The pKa is the pH at which a drug is 50% ionized. Understanding pKa is essential because it helps predict how the drug will behave in different environments, such as the stomach or intestines. It also guides the selection of excipients such as buffer and determines the pH range at which the drug is most stable. pKa is also crucial when designing the dosage forms that rely on the drug's solubility and permeability. Question 8. What is hygroscopicity and why is it a concern? Hygroscopicity refers to a drug's ability to absorb moisture from the surrounding environment. This property can lead to degradation, affecting both the stability and the efficacy of the drug. It can also cause changes in the powder's flow properties and compressibility, making it harder to process. Packaging and storage conditions are the critical parameters preventing moisture absorption in hygroscopic drugs. Question 9 is, why is bulk density measured during preformulation? Bulk density is a measure of the mass of a powder divided by its bulk volume, including void spaces. It helps us understand the flow and packaging properties of the drug powder, which are essential for designing tablets and capsules. Bulk density also affects the uniformity and consistency of the dosage form. This measurement is important in determining how much of the drug can be packed into a given volume. Next question is, how do you check drug excipient compatibility? To check the drug excipient compatibility, we perform several studies using techniques such as DSC, FTIR, and HPLC. These methods helps us to detect any physical or chemical interactions between the drug and excipients that could lead to the degradation. Ensuring compatibility is crucial for maintaining the stability and effectiveness of the final formulation. Any adverse interactions need to be identified early in the processes. Next question is, what is role of stability studies in preformulation? Stability studies are designed to assess how a drug behaves under different environmental conditions such as temperature, humidity, and light exposure. These studies help us to determine the drug's shelf life, storage conditions, and whether the drug remains stable throughout its intended use. They also identify degradation products and their potential effects. Stability testing is essential for ensuring the quality and safety of the drug over time. Next question is, what is the difference between intrinsic and apparent solubility? Intrinsic solubility refers to the solubility of the unionized drug in a solvent, which remains constant regardless of pH. Apparent solubility, on the other hand, includes both ionized and unionized form of the drug and can change with pH. Apparent solubility is more relevant when considering the drug's behavior in the gastrointestinal tract, where pH varies. Understanding both solubility types helps in optimizing drug formulation for better absorption. Question 13 is, how does particle size affects formulation? Particle size plays a significant role in determining the drug's dissolution rate and bioavailability. Smaller particles have a larger surface area, which enhances their ability to dissolve quickly. However, reducing particle size may affect the flowability and compressibility of the powder, which is important in tablet or capsule manufacturing. Therefore, particle size must be optimized to balance solubility and processing properties. What is the difference between amorphous and crystalline forms? Crystalline drugs have a well-defined molecular structure and are typically more stable but less soluble. Amorphous form lacks their organized structure and tend to have higher solubility but are less stable. The choice between these two forms depends on the drug's intended use. Amorphous forms are often preferred over drugs that need to be absorbed quickly, while crystalline forms are chosen for stability. Question 15 is, how is preformulation data useful for regulatory submissions? Preformulation data is a key component in regulatory submissions such as ANDA or NDA filings. It provides scientific evidence to support the chosen formulation, dosage form, and manufacturing process. This data also helps demonstrate that the drug is stable, effective, and safe. Regulatory agencies require detailed pre-formulation studies to assess the drug's quality and ensure that it meets the necessary standards. Question 16 is, what is dissolution rate and why it is important? The dissolution rate measures how fast a drug dissolves in a given solvent, which is crucial for the absorption and bioavailability. A faster dissolution rate can lead to quicker onset of action, while a slower rate may delay therapeutic effects. By optimizing the dissolution rate, we can ensure that the drug reaches its therapeutic level in the bloodstream more effectively. It is an essential parameter in ensuring constant drug performance. Next question is, what is the role of surfactants in preformulation? Surfactants are compounds that reduce the surface tension between two phases such as water and oil, improving the solubility of poorly water-soluble drugs. They can enhance drug dissolution and bioavailability by improving wetting properties and reducing interfacial tension. Surfactants also stabilize formulation by preventing particles from aggregating. Proper selection of surfactants is crucial for optimizing drug absorption and formulation stability. Next question is, what is the significance of melting point in the preformulation? The melting point is an important physical property that can help us to identify the property and stability of a drug. A narrow melting point ranges indicate a high level of purity, while a broad range suggests impurities. It can also provide insights into the crystalline or amorphous nature of the drug. The melting point is critical for ensuring the quality and consistency of the API. Next question is, what are the challenges in the preformulation studies for poorly soluble drugs? Poorly soluble drugs face challenges in absorption, which leads to low bioavailability. We may need to use techniques such as solid dispersion, particle size reduction, or the addition of surfactants to improve solubility. The formulation processes also become more complex, requiring careful excipient selection to ensure stability and uniformity. Optimizing these factors is crucial to overcome the limitations of poor solubility. And the last question is, how do you assess the compressibility of a powder? Compressibility is assessed by evaluating the change in volume when a powder is subjected to pressure. It is typically measured using parameters such as Carr's index and Horner's ratio, which help determine how easily the powder can be compressed into tablets. Poorly compressible powders may lead to inconsistent tablet weight or poor tablet hardness. Ensuring good compressibility is very much essential.